I'd like you to imagine doing something we all do on a daily basis, which is flipping a light switch. Ideally, the light simply turns on, but every once in a while, the circuit may be overloaded or the switch may be faulty, causing the light to flicker. In other words, the electrical activity is unregulated. Now I'd like you to imagine these unregulated electrical impulses occurring in the brain. You might be imagining a seizure in which abnormal and excessive electrical activity is spread from neuron to neuron. Seizures may cause uncontrolled muscle movements and can bring about temporary changes in a person's consciousness and memory. Now, a lot of us probably associate seizures with epilepsy, but seizures themselves don't always mean that a person has epilepsy. Isolated seizures may be provoked when the body is stressed, like with a high fever or following a brain injury. Seizures become classified as epileptic when they recur in an individual with a predisposition for seizure generation and a lower seizure threshold, meaning something that's relatively innocuous to someone without epilepsy might generate a seizure in someone who does have epilepsy. Photostimuli, like flashing lights, for example, may trigger a seizure in someone who has epilepsy. There are many different epilepsy syndromes, and most of them are caused by mutations in a variety of genes, which ultimately impacts electrical signaling in the brain, leading to this predisposition to seizure generation. In our lab, we study genetic mutations encoding neuronal proteins, which are responsible for the generation and propagation of electrical signals. Alterations in these proteins may disrupt normal patterns of electrical signaling in the brain, leading to uncontrolled electrical activity and epilepsy. Many of these mutations are known to cause epilepsy syndromes that impact newborns and infants, and their seizure activity is highly resistant to treatment with anti-seizure medications. Epileptic activity disrupts brain development, leading to cognitive and behavioral impairments. Often, developmental milestones aren't reached, or they are reached and then are lost following the onset of seizures. Even if the seizures naturally subside over time, the delays and regressions are permanent. This can lead to children being unable to speak, unable to execute motor activities, even as simple as sitting up, and in severe cases can be fatal. The devastating impact of these mutations is why it's so important that we study them. For some illnesses, we can biopsy and examine tissues, but we ethically cannot biopsy brain tissue, especially in individuals who are so young. So how do we go about studying these mutations, especially if they're uncharacterized and they're not yet known to underlie an epilepsy syndrome? Genetic testing can identify whether a mutation is present in one of these genes, but it can't tell us how neuronal functioning is impacted and whether the mutation is capable of causing epilepsy. What we can do is examine the effects of these mutations in a two-step process. First, we can use molecular biology and physiological techniques to determine the effect on electrical signaling. Then we can build computational models of neurons incorporating numbers generated from the first step. This allows us to simulate electrical signaling in neurons that express these mutated genes relative to healthy neurons. To achieve this, we use a program called Neuron in conjunction with the programming language Python. Neuron was developed to help scientists build the complex morphology of neurons. It can be used to simulate signal transmission and integration in the nervous system. Since neurons generate and propagate electrical signals, sections of a neuron can be modeled as individual electrical circuits, whose properties are represented by cellular structures. Acting as a capacitor, the cellular membrane can store charge. Proteins embedded within the membrane can act as variable resistors and may block or conduct the flow of current under certain conditions. This current is provided by electrically charged ions, like sodium, potassium, chloride, and calcium, which are found inside and outside of cells. This separation of charge provides the neuron with a voltage and acts as a battery to drive the flow of current across the membrane when permitted. When we apply a stimulus to the model, the capacitor charges, the conductors activate, and ionic current flows across the membrane. This changes the neuron's voltage over time and generates a waveform like this. This waveform represents the electrical signal that was generated by a neuron in response to receiving a stimulus from a neighboring neuron. This stimulus may propagate down the length of the cell to trigger the release of chemical neurotransmitters, which may stimulate the next neuron to generate its own electrical signal. When we incorporate a genetic mutation, a neuron's response to stimuli changes. If a mutation does contribute to the symptoms of epilepsy, we would expect to see changes in the timing, amplitude, and frequency of the resulting signal. A stimulus that produced a single waveform in a healthy cell may generate multiple spikes in a cell with an epilepsy mutation. This increased frequency of electrical activity is characteristic of brain activity during epilepsy. Even if we learn that a mutation doesn't impact neuronal functioning in this way, we still benefit from the simulation. If we know what isn't causing these symptoms, we're still a little closer to discovering what is. Neuronal models aren't just limited to studying epilepsy. For starters, they can be used to examine brain activity in other pathologies, like Parkinson's disease or depression. But it's also important to remember just how complex the brain is, that there's still so much that's left to be discovered, even about healthy functions, that can be explored using computational models. Neuronal models can show us how individual neurons communicate with one another, or how thousands of cells function in complex processes like respiratory control in the brainstem, sleep-wake cycles, and working memory. They can be used to simulate physiological events like changes in electrolytes or body temperature to see how neurons function under different conditions. All of this can be achieved without having to collect human tissue samples. Computer simulations provide the foundation for advancing our knowledge of pathologies like epilepsy. Since there are so many underlying genetic causes, each with their own pathological mechanism, that means there is no one-size-fits-all treatment. If we want to develop a therapy for severe treatment-resistant syndromes, like those impacting newborns and infants, then we need to understand the problem. We need to understand what cellular process is being impacted by a particular mutation. Once we know what is going wrong, then we will know what needs to be treated. Using these tools, we can provide a therapeutic pathway to either prevent these symptoms from occurring at all or to reduce their negative impacts, so we can provide individuals with a better quality of life.